Okay, can you all hear me? Can you hear me?
Yes.
Okay. I'm going to start sharing my screen. Can you see my screen now? Hello? Can you see my screen?
Yes.
Okay, very good. Now we will start solving past papers. You all need to practice paper 4. Like all the grades were not very good. Paper 4 were the lowest grades, and this is normal. Always you lose most of the grades in paper 4. And paper 4 is the most important paper, the one with the most weight. It's 80 marks. So your exam, I think I sent you the dates. The first exam is paper 4, which is nearly in a month, like a little bit more than a month. So you need to focus on paper 4 and year 10 notes. Okay, now let's solve the 2023 exams. This is paper 41, June 41, last year exam. So your friends last year, they solved this exam. This exam, you'll find it on the site that I sent you. Okay, let's start and not waste any time. So you can all see the screen and you can all hear me clearly, yes?
Yes.
Yeah.
Okay, very good. Now we'll start immediately. Now tell me, 1A, figure 1.1 is the diagram of the digestive system. Each letter may be used once, more than once, or not at all. State the letter of the part shown in figure 1.1. What is the part that produces bile? Give me letters.
K.
Yes, sure. It's K. Give me a second. Yes, K. So K is the liver. It produces bile. Now, quickly, what's next? So that produces bile. This is K. That produces gastric juice. What is gastric juice? Juice produced where?
Pancreas.
No, no, no. Gastric means stomach.
Oh.
Stomach, okay? Gastric juices. So where will this be? The gastric juices, where are they? Is this going to be?
C.
C, okay? Good. So gastric juice is C. And which part produces urea? Produces urea, not excretes urea.
Liver.
Very good, liver, which is also K. Where maltose is digested. Maltose is digested. Where's the picture? Maltose, maltose, not starch. So where's the answer?
H.
Yes, correct. Maltose is digested in the small intestine. In the mouth, in L, L is the oral cavity or the mouth, and A are the salivary glands. Starch is broken down into what? Maltose. And then the second stage in the breaking down of carbohydrates is in the small intestine, in H, where maltose will be broken down into glucose. So the answer is going to be H here. Where trypsin acts, where is trypsin?
Stomach.
Mm. No, no, no.
Full intestine.
Small
Trypsin is
intestine.
in the small intestine, in H also. So trypsin is a protease. It breaks down protein into amino acids, and trypsin in the small intestine. The enzyme in the stomach is the pepsin. Pepsin. So don't get confused. This is supplement, by the way. In the supplement, there are two additions. Number one, addition one, where are carbohydrates broken down in the digestive system? Carbohydrates are broken down in two parts, in the mouth and the small intestine. In the mouth, starch is broken down into maltose, and in the small intestine, continuation of digestion takes place, where maltose will be broken down to glucose. Okay, this is the small intestine, or this is the digestion in terms of carbs. In terms of protein, let's revise together. The protein is not digested in the mouth, only in the stomach and the small intestine. What is the enzyme that digests protein in the stomach?
Pepsin.
Pepsin.
Pepsin, good girl. And what is the name of the enzyme that digests protein in the small intestine?
Trypsin.
Trypsin. Well done. Okay. Now, paper 4, don't be surprised if paper 4 gives you an investigation. They actually always give you an investigation. Okay? Let's read it together. A student investigated the effect of bile on the digestion of fat in milk. They set up 3 different test tubes. Test tube A contained milk and bile only. And test tube B contained milk and lipase enzyme only. And test tube C contained milk and lipase and bile. Now, before I move on, which test tube will show me the fastest digestion of fats? A, B, or C?
C.
C, of course, because it contains everything needed for the digestion of fats. They used an indicator that is pink in alkaline solutions and colorless in acidic. Okay, they added the same volume of indicator to each test tube. The student observed and recorded the number, the color of the contents of each test tube at 0 minutes, 20 minutes, and at 40 minutes. And the table shows the results of the investigation. So test tube A, B, and C at 0 minutes, at 20 minutes, and at 40 minutes. Pink, they said pink, this is when it is alkaline, and colorless, this is when it becomes acidic. Okay, now explain the results for test tubes B and C in table 1.1. B and C. There is always a question like this in paper 4 exam. In the mock exam, they got you the antibiotic sensitivity test, if you remember, and they also told you to compare. So there is always an investigation question where will you have to explain your results. Explain means describe it, write reasons, give numbers from the table, etc., etc. Now, how do you answer a question like this? They clearly want you to explain the results for test tube B and test tube A. So you can divide it into test tube B and test tube C. Let's go back to the question. Test tube B, what was in the test tube? Milk and lipase only. So you only have milk and lipase. Test tube C, milk plus the bile plus lipase. Okay? In test tube B, let's look at the results. We started with pink color. So at 0 minutes, it was pink. And then after 20 minutes, it was still pink. After 40 minutes, after a very long time, it became colorless. Can you explain why? Hmm? Why did it change from pink to colorless in test tube B? I have milk and lipase enzyme. Give me an answer. I will not answer every single question for you. Hello?
Contain fats.
Yes, it contains fats, and these fats were broken down by the lipase enzyme to give me what?
Fatty acids and glycerol.
Very good. Fatty acid and glycerol mentioned that. And the fatty acids, these are acidic, so they will be responsible for the color change. And it took 40 minutes. Put this in concentration. Let's look at C. It contained the milk and the bile and the lipase. And at C, at 0 minutes, it was pink. In other words, it was alkaline. After 20 minutes, in half the time, it became colorless. So only after 20 minutes, it became colorless. Can you give me an explanation for that?
There's more enzyme, it's stronger.
No, there is not more enzyme. There is something extra. In C,
Catalyst,
I had excuse me?
a catalyst,
No. The lipase was present in both test tube B and test tube C. What is extra in test tube C?
Bye.
The bile. Yes, test tube C contains the bile. Bile is the added component. And what does bile do? What is the function of bile?
Emulsification of fats.
Very good. You will explain that. Bile is added in C. Bile will help in the physical breakdown of fats or the emulsification of fats and describe it, which is the physical breakdown of fat from large particles to small particles. And why is it important?
It increases the surface area for
Correct.
enzymes to work faster.
Correct. Perfect. So you will mention the function of bile, physical breakdown of fat, which is called emulsification, that will break down fat from large particles to small particles, and this will increase the surface area. That is why the reaction was faster in B or C.
C.
In C, of course. Okay. So this is the explaining part. You will take B and you will discuss, and you will take test tube C and you will discuss, and you will have to compare. And then you will have to explain why is one different than the other. Okay. So again, at B, it will change from pink to colorless because fat will be broken down by the lipase enzyme, but it took me 40 minutes. In test tube C, it will change from pink to colorless in 20 minutes, in half the time. So in test tube C, the reaction was faster. Why? Because I have bile added to test tube C, and bile will make the reaction faster because bile will do emulsification and describe it, and bile will increase the surface area that will help the enzyme lipase to work. Okay. And why does it become colorless? Because it becomes acidic, because fatty acids are acidic and they will reduce the pH. The pH will be decreased. You have to mention everything that crosses your mind. Everything that crosses your mind. Okay, in order to take the 4 marks. Also, most of you in the mock exam did not compare every single part in the Petri dish because you just looked at the one with the most clear zone and you ignored the rest. So most of you lost a mark or two in this question. So is this question clear? Do you understand how you will break down this question? Any questions before I move on? Okay. Explain the purpose of test tube A in table 1.1. Let's go to the table. Test tube A. Test tube A, let's go to the information. Test tube A contains milk and bile without the lipase. What is the importance of test tube A?
Control.
Control.
Okay, this is not enough to say the word control. These are 2 marks. Control, then what? Why do we have a control?
To compare the
Okay.
results.
Comparison.
Very good. Again, this is not enough. Control for comparison. I want to compare what?
The effect
A
of
contains,
lipase on the
excellent,
rest of the enzyme.
excellent. Test tube A contains milk and bile only without lipase. In the other test tubes, I have lipase. So I want to know if the lipase will affect the reaction or not. I want to know the effect of lipase. Okay? So control, to compare. Again, this is not enough. The problem with biology is that usually things are not enough. I want to know if the lipase is going to affect the reaction or not. Okay? If the lipase is needed for the breakdown of the fats or not. Now, this exam is focusing on the lesson enzymes. The action of lipase is affected by temperature. Figure 1.2 shows the axes for a graph of the effect of temperature on the activity of lipase. Complete the graph by drawing a line to show the effect of temperature on the activity of lipase, and then adding a label line and a label to show the point at which all the lipase has been denatured. Now, I think you are very familiar with this graph. How will the graph look like? Come
A
on.
curve down.
It will look like a hill like this. Okay? And then here, draw a line to show the effect of temperature. And then we will add a label line and show the point at which all the lipase has been denatured. Which point? A, B, or C? A or B or C?
See.
C.
Of course, I'll make here a line and I'll give them what they want. Very easy question. This is an easy question. Explain why lipase cannot be used to catalyze the breakdown of proteins. This is a nice question here.
Because lipase is responsible for the digestion of fats, not proteins.
Okay, you will take 1 out of 3. We are talking about enzymes in general.
Oh, they don't have the same. They don't have complementary active site to the substrates.
Of course, enzymes are specific. Enzymes are specific. And you will mention what you just said, that enzymes have a specific active site that is complementary to a specific substrate. And you will mention the example that you have in the question. For example, the lipase will only act on fats to break it down into fatty acid And glycerol, it cannot act on proteins because they have a specific or a different shape. Okay. And when the enzymes bind with the substrate, they form an enzyme substrate complex. This is not possible with lipase and proteins. Okay. This is called the lock and key mechanism. Look how many keywords. Specific is a keyword. Active site is a keyword. Complementary in shape is a keyword. Lock and key is a keyword. Okay. Please, please, please don't just write one sentence and leave. Okay. Up to now, I see that it's very easy. Question 2. A student investigated osmosis in potato plant cells. He immersed cubes of potato tissue in water and different concentrations of sucrose for 30 minutes, and the mass of the potato cubes was measured before and after. The percentage change in the mass were calculated, and table 2.1 shows the results. I assume that you are very familiar with that. Look at the table. You have different concentrations here. Mass of the potato at the beginning, mass of the potato at the end, and the percentage change. Of course, they will tell you to calculate the percentage change here. How will you do it? Percentage change.
Final minus initial
Final.
over initial times 100.
Yes. Okay. And the answer will be negative. This is normal, like the other two answers. Read the question carefully. Use the information in table 2.1 to calculate the percentage change in mass at 1 point at here, when the concentration was 1 mole per decimeter cubed, and give your answer to 2 decimal places. Now, I want you to look here. Space for working. Space for working, please show your steps. Don't just give me the final answer. Okay, there's always a grade for working. So you will show them clearly. 1.11 minus, this is the final, minus the starting, 1.28 over the starting, 1.28 times 100. Okay, let me calculate it. 1.11 minus 1.28. This is minus 0.17. Please do this step on its own. Divided by 1.28. This is going to be minus 0.1.28, excuse me, 0.17 divided by 1.28. Don't forget times 100. It's going to be minus 13.28. Okay. You want us to open the mark scheme? Let's open the mark scheme. 1 minute. Which number? This was number 2. Yes, minus 13.28. Okay. You have to show your steps. What's this? Oh, this is not what I want. Okay. Now, using the information in table 2.1, explain the difference in the results between 0.6 moles per decimeter and 0.8 sucrose solutions, and use the term water potential in your answer. How many marks? 5 marks. So let's go and compare. We want to compare here the results between 0.6 and 0.8. Let's look at 0.6, break it down, and 0.8 and break it down. At 0.6, the starting mass was 1.33, final mass 1.29. These numbers are what important? The percentage change in mass is minus 3.01. When it was 0.8, the percentage change in mass is minus 8.2. Okay, what will you say about 0.6? What will you say? You will nearly say the same thing, by the way. So what will you say about the concentration? What happened to the potato at concentration 0.6? This is the potato, and it was put in sucrose solution that is of 0.6 concentration. And the potato decreased in mass. What will you say?
Flaccid.
Water
Okay, this
moved
is the
out
only
by
thing you will
osmosis.
write. Can you please elaborate? You have 5 marks.
Water moved by osmosis out of the potato.
Yes, you are correct. But they said that we want you to use the word water potential.
Okay, so
High
to
water
move from high water potential to low water potential outside the potato.
Okay, so the water potential was high inside the potato or outside the potato?
Inside.
Of course. So you will mention what you will use the word water potential because they asked you to use it. So the water potential was higher inside the potato and the water potential was lower outside the potato. So this will make the water move by osmosis down a concentration gradient from the inside of the potato to the outside of the potato. After you say all that, you will say that the vacuole will shrink and the cell membrane will detach away from the cell wall and the cell will be flaccid. Okay, there was someone here who jumped to the last part of the answer. She told me flaccid. Okay, so here you're jumping to the last part of the answer. You have to explain how did it become flaccid. You will mention this here. And by the way, you will mention the same case when the concentration was 0.8, but you will add something extra. What will you say? When it was more concentrated, when the potato was in a sucrose solution that is of 0.8 concentration sucrose, it was very concentrated. You will say the same thing, but you will just add something, one different word. What is it?
Turgor pressure.
No. We will also say that. You will simply say that the water potential of the sucrose solution was lower or higher than the 0.6. It's more concentrated or less concentrated. Why is this hard for you? I have two situations, a potato in a concentration of 0.6 and a potato in a concentration of 0.8. And in both cases, the potato decreased in size. So in both cases, we are talking about flaccid or plasmolysis. You told me in the 0.6 that the water potential here was higher and the water potential outside was lower, and this is correct. And the water will move by osmosis from the inside of the potato to the outside of the potato. Isn't this the same situation in the 0.8 concentration?
Yes.
The water potential is higher inside the potato and it is lower outside the potato, and the water will also move from the inside to the outside. Yes, but we are comparing. What is the difference here?
Concentration
There is a
gradient.
Excuse me? The concentration gradient. Yes, there is a bigger concentration gradient. Or in other words, you will say that the 0.8 concentration is very concentrated and it is of a lower water potential. We have even less water here in this example, and more water will move out of the potato by osmosis. That is why the potato here will shrink more. So here, in both cases, water will move out. But in example A, less water will move out. And in example B, when the surrounding sucrose solution was more concentrated, more water will move out by osmosis. That is why it will shrink more. It will be more flaccid. Did you understand? So the situations in both of them are the same. Both of them, the potato is in a concentrated solution, but one of them is more concentrated than the other. That is why the potato will shrink more than the other because more water will move out. Is this hard? Hello? Is this hard? The problem is that you understand things while we explain it together, but I don't know why it does not cross your mind on the exam. Okay? When they tell you to explain the difference between one investigation and the other investigation, then you have no choice except to explain the difference. Whether you like it or you don't. You will not talk about one and ignore the other. You will have to talk about both and you will have to find a difference because you are asked clearly to explain the difference. So you will say that this showed flaccid and this showed also plasmolysis. They're the same thing. What's the difference? What can you say? You have to find the difference. Which one lost more water, the 0.6 or the 0.8?
The 0.8.
The 0.8 because it was more concentrated. Okay, because you want the 5 marks. Let's look at the mark scheme. Okay, here is the mark scheme. The mark scheme is very clear. You have to mention the difference, the only difference between them, which is one of them, more water will be lost. More water leaves the potato tube that is more concentrated, the 0.8 mole per decimeter. Do you understand where the problem is? You understand things when we explain it together, but I don't know why sometimes just things don't add, don't cross your mind. One is in time to change color. It's not trying to change color. Okay. Here now, better? Okay, let's not waste time on this question. Describe the expected appearance of a cell from a potato cube that has been immersed in distilled water for 30 minutes. So how will you describe this cell? In distilled water, distilled water. What does distilled water mean?
Pure.
Yes, very good. You have to know the names: hypertonic, hypotonic, concentrated, dilute, distilled. Distilled water means that the potato was put in pure water for 30 minutes. How will this potato look like? One cell from this potato, how will it look like?
Turgid.
Very good. It will be turgid. This is an explanation. Explain. How does the turgid look like? How does the turgid cell look like? Talk to me about the vacuole. The vacuole will shrink or swell?
Swell.
Swell.
Of course, the vacuole will swell. It will be swollen. The cell membrane will be detached or will be pushed against the cell wall.
Pushed.
Pushed.
Yes, it will not be detached. The cell might bulge a little bit. It will be turgid. You're correct. It will bulge a little bit. Please write more. Don't just write one word and leave. Describe how the process of active transport differs from the process of osmosis. I think this is very easy, and this question came on your exam. How do you compare between active transport and osmosis? This is a core question. Can you be quick?
Osmosis is only used for water.
Yes, only water. Very good. Active transport for any molecule or any particle. What else? I want 3 differences.
Active transport happens from low to high concentration, while
Correct.
osmosis
From
happens
low to high.
while
I
Osmosis
want
has a
another
special
difference.
membrane.
Yes, active transport needs a semi-permeable membrane.
active transport needs
Actually,
carrier
osmosis
proteins.
also needs a semi-permeable membrane. This is not the difference. It needs carrier proteins. Yes,
Needs
active
energy
transport
for
needs carrier
respiration.
and needs energy. Very good. Okay, osmosis does not need carrier proteins and does not need energy. State the type of plant cells that use active transport to absorb mineral ions from the environment.
Root hair cells.
Correct. Yes, this is easy. Root hair cell. Explain the effect of the lack of magnesium on the color of the plant leaves. 2 marks. Magnesium is
Magnesium.
needed to make what?
Chlorophyll.
Yes, chlorophyll. Now, when there is no chlorophyll, don't just write
Yellow leaf.
chlorophyll.
Like
Yes,
yellow.
there will be no photosynthesis. No photosynthesis, the leaves will be
Become
yellow.
yellow.
Yes, it's called chlorosis, by the way. Chlorosis. Okay, this is enough. Okay, easy exam. Number 3, figure 3.1 is a photomicrograph of some cells lining the trachea. And this is a goblet cell. Describe the role of the goblet cell. 2 marks.
Secrete mucus.
Easy question.
With mucus.
Yes, of
This
course.
is mucus.
Yes, 1 minute. Yes, of
Has-
course, it secretes mucus. And what is the function of the mucus?
Trap dust.
Dust
Bacteria.
Particles.
Yes, very good. It will trap dust and trap pathogens or bacteria. And that's it. This traps dust and protecting of the respiratory tract. Explain how the cell labeled X in figure 3.1 is adapted for its function. What is cell X? This cell.
Cilia.
Very good. This is a ciliated cell. Actually, it is called the ciliated epithelium. Okay. And what does it do?
It pushes the mucus away from the
Sweeps away
away from
dust.
the lung.
Yes, the word sweeps is better. It doesn't push, it will sweep. Okay. It will sweep the mucus, which contains bacteria, away from the what?
The
lungs,
Of course, away from the lungs in order to prevent what?
coughing.
To prevent infection.
Blockage of the
Okay. To protect the lungs. Correct. Very easy. State the name of one other part of the body where the type of the cell labeled X in figure 3.1 is found.
Small
Where
intestine.
is ciliated?
Small intestine.
No, what is
Bronchi.
found in the small intestine is the microvilli, not cilia. Bronchi,
الألفيولاي اللي هي في
not
اللنجز.
the alveoli, the bronchioles, the bronchi and the bronchioles, also in the small intestine. And also, by the way, we took it in the oviduct. This is in year 9 notes. If you remember, the oviduct contains cilia because when the egg is released from the ovary, this cilia will push the egg towards the uterus. So it is either the bronchi or bronchioles or the oviduct. Okay. Table 3.1 shows some features of the breathing system. Complete table 3.1 to show the actions of each feature of the breathing system that occur during inspiration. What happens to the diaphragm when you breathe in?
contract becomes
Contract.
flat.
Very good. It will contract. Yes, it will contract. What will happen to the external intercostal muscles?
Contract.
It will move up and
Also
outwards.
contract. We said they were friends. Okay. Pressure in the thorax. 1 minute.
Decrease.
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 1 minute. The pressure, what happens? Yes, the pressure when you breathe in will increase or decrease?
Decrease.
It will
Decrease.
decrease, correct. And the ribs will, how will they move?
Move up.
Move.
Up and
Excellent.
out.
Up and out. Excellent. And the volume?
Decrease.
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 No, not decrease. It will increase.
Increase.
Yes, correct. Very good. Okay. If you did not study the lesson respiration, you will lose 5 marks in this question. State the name of the gas that is excreted by the breathing system. Now, this is so easy.
Carbon
Carbon
dioxide.
dioxide.
Carbon dioxide.
Very good. If you give me the name of another gas, what else is
Vapor.
excreted by?
Water vapor.
Excellent. Yes. Very good. H2O gas, water vapor. Okay. Good ventilation is one feature of the gas exchange surface. State 2 other features.
Rich blood
Surrounded
supply
by
and large
capillaries.
area.
Yes, I think I heard you. Rich blood supply. Correct. What else?
Large surface area.
Yes, you are correct. Large surface area. Also, it has thin walls. It is moist for the dissolving of the gases. Any of those. Very good. This is a very easy exam. Say the name of the gas exchange surface in humans.
Alveoli.
Correct. Perfect. Alveoli. Okay. Now, this is plants. Figure 4.1 shows the effect of light intensity on the rate of photosynthesis at different temperatures and different concentrations of carbon dioxide. So you have the light intensity on the x-axis, rate of photosynthesis on the y-axis, and you have A, B, C. Now, this describe and explain the reasons for the shape of the line B and C. Okay, unfortunately, you are not good at these questions. This is one of the questions that also came in the mock exam, the Petri dish question, and you did not also explain things well. This question is very obvious, or this style of questions, if you notice, more than once it can come on the same exam. So here you will describe and explain the reasons for the shape of the line B and C. Okay, any graph, we will break it down. I will not help you. Can someone tell me what will you say for graph B?
It has higher CO2 than C.
It has higher what?
Carbon dioxide.
This is all what you will say about B?
Higher rate of respiration تيجي.
No. I'm very upset that you don't know how to break down those graphs. Look, the first thing you will say about B is that you will describe the shape. What is happening at the beginning? And then
Increases,
what is
then level
happening?
up.
As the light
Yes.
intensity increase.
Okay, you will describe the graph and then you will explain it. Describe it means what? At the beginning, as the light intensity is increasing, what is happening to the rate of photosynthesis?
Increasing.
It is also increasing. We said we will always break the graph into 2 or 3 parts depending on its shape. This graph, I'll divide it into part A and part B. So at the beginning, I will say that as the light intensity is increasing, the rate of photosynthesis is increasing. And then what will I say at B?
It stays constant.
Levels off.
Yes, as the light intensity is increasing, the rate of photosynthesis will become constant. Okay, this is the describing part. If I had numbers on the graph, I will also give numbers. Now I will go back and explain A. At A, I said, as the light intensity is increasing, the rate of photosynthesis is increasing. Now, why? What is the limiting factor at A?
مفيش حاجة.
How
Current
come?
Light intensity.
No, the light intensity itself. You forgot the lesson. At A, every time I'm increasing the light intensity, the rate of photosynthesis is increasing. So this is showing me that the more I give light, the more the photosynthesis. So this is showing me that at A, the light intensity is the limiting factor. The limiting factor, which is the factor that will stop the rate of photosynthesis from reaching its maximum. Now, the second part of the graph, when the light intensity is increasing, but the rate of photosynthesis is constant. Is the light intensity a limiting factor in this part of the graph?
No.
No.
No, it is not. Then something else must be a limiting factor. What is the limiting factor at B?
Temperature or CO2 concentration.
Exactly. Exactly. Temperature, because B was at 15 degrees Celsius and at A, well, it was 25 degrees Celsius when the rate of photosynthesis increased. Okay, so this is what you will say at B. Again, this is 6 marks. And I don't know why you don't break things down even though you know it and you took it before. So let's go back to B. I will describe and explain. Describe means I'm going to describe the shape of the graph. And if the graph has numbers, I will write numbers. So at B, this is B. The shape of the graph looks like this. It increases and then decreases. So I'm going to break it down to 2 parts, part 1 and part 2. Part 1, I will say, as the light intensity is increasing, the rate of photosynthesis is increasing. Finished. Part 2, I will say, as the light intensity is increasing, the rate of photosynthesis becomes constant. So I described. Now I will explain. In part 1, or in the first part of the graph, when the graph is increasing, why is it increasing? What is the limiting factor?
Light.
The light intensity, whatever is on the x-axis, because there is proof. Every time I give light, every time the photosynthesis is increasing. So in the first part of the graph, the light was the limiting factor. In the second part of the graph, the light is no longer a limiting factor. The limiting factor has to be something else. What is it? I will look at graph A. I will see that the temperature became higher. So in this part of the graph, the limiting factor was the temperature, not the carbon dioxide concentration, because the dioxide concentration was the same. Did you all understand how did we explain B? Does anyone have any questions? Now let's go to graph C. I still did not finish the question because they want you to compare B and C. Let's go to C. Again, graph C is divided into 2 parts, the first part and then the second part. In the first part, as the light intensity is increasing, the rate of photosynthesis is increasing. So what is the limiting factor here?
Light intensity.
Light intensity. Now, the second part of the graph, as the light intensity is increasing, the rate of photosynthesis is constant. So it is no longer a limiting factor. What is the limiting factor at C?
Temperature.
No, the temperature, yes, the temperature, you can say that, because when we increased it to 25, the rate of photosynthesis increased. What else?
Oh, the CO2 concentration.
the carbon dioxide concentration. That's it. That's it. Okay? And it's clearly here. It's in your notes somewhere. I don't know where is it, but it's there. Yes, here, effect of temperature. In this part of the graph, the temperature was the limiting factor. And this always in the increasing part of the graph, this is the light intensity. This is exactly the question. Just need to study more. Okay. This is one of the products of photosynthesis. What is this?
Glucose.
Glucose. What is the chemical formula for the other product?
Oxygen.
Water.
O2.
Yes, O2.
God.
Okay, I'm sorry. I just need to, I'll come back in 1 minute. Just be there. Okay, I'm back. I'm sorry. Now, outline how the carbohydrates made during photosynthesis are used in plants. Okay, this is a very nice question. This is core, by the way. What is the carbohydrate that is produced in photosynthesis?
Glucose.
Glucose, very good. And this glucose, it is used in what? How will the plants use the glucose?
It's converted to
Give me
glucose.
4 uses of glucose in plants.
Converted into
Cellulose.
cellulose to make cell walls.
Very good. I'm going to convert it into cellulose to make cell wall. Another use.
Starch.
For respiration.
I will store it as starch. Very good. Yes, I will store it as starch. Okay, for storage.
Imported as
And very good. Excellent. I'm going to convert it into sucrose for translocation to distribute it in the flowing from the source to the sink. And another or a final use for glucose. Very easy one.
to make nectar.
Nectar.
Yes, very good to make nectar. And of course, I can use it to make energy in respiration. This is a very easy question. This question is core. Very easy question. Okay. Now this is protein synthesis. Figure 5.1 shows the stages involved in protein synthesis. State the names of the parts labeled X, Y, and Z. What is X?
Nucleus.
Okay. No. Yes, yes, it is the nucleus, but yeah, okay. Nucleus.
Nucleus 4.
Yes, it's correct. What's Y?
mRNA.
mRNA.
RNA.
Where is Y? Oh, yes, correct. Messenger RNA, good girl. What is Z?
Ribosome.
Correct. Ribosome. Now this is supplement for sure. State what determines the sequence of the amino acids in the protein that is produced.
The genes that are
Okay.
the sequence.
Someone
sequence
said
of
it. I want
bases?
a better answer. Huh?
The sequence of bases?
Correct. The sequence of bases where?
And the mRNA.
RNA.
Yes, in messenger RNA, which originally came from the DNA. So just mention that it's in the sequence of bases in the messenger RNA or in the DNA. Explain
Okay,
why the
gene
sequence
correct.
of amino acids, excuse me?
Is gene correct instead of basis?
Messenger RNA is even better. Yes, of course it is correct. Explain why the sequence of amino acids is important in the production of receptor molecules for neurotransmitters. Now, this is a tricky question. Explain why the sequence of amino acids is important in the production of receptor molecules for neurotransmitters. Now, when the
Think of the
neurotransmitters are made up from amino acids, then what are they made up of?
proteins.
Proteins.
Protein. Now, the sequence of amino acids, why is the sequence important?
So they have a specific shape, so that the substrate can fit in.
Excellent. The sequence of amino acids will determine the final shape of the what?
The receptors.
Very good, of the protein, which is the receptor. And the receptor, they have a specific shape, okay, to bind with their neurotransmitter. They bind with a specific neurotransmitter. They have to be, please use the word complementary. They have a complementary shape, okay? And don't write enzyme substrate complex. It's not an enzyme. It's just specific, and the word complementary will give you the 2 marks. Very good. Explain why body cells can have different specialized functions even though they contain the same genes. Again, this is a very important question. We took it in inheritance.
Only some genes are expressed.
Excellent. Very good. And this is called gene expression.
Expression.
Excellent. Body cells, all body cells contain the same genes. So you will start by saying that all body cells, they contain the same genes. But just like you said, not all genes are expressed, only some, only the genes that are needed, okay? Only needed genes or the needed proteins are expressed. And if they ask you for the advantage, this is called gene expression. What are the advantages of gene expression? Why are some genes expressed and not others?
Allows specialization.
Yes, it will allow specialization and it will save. Hmm? It will save energy. It will save resources. I don't need all those genes. Only specific genes are expressed or activated. Now, allele frequency in a population can be changed by natural selection and artificial selection. State the meaning of allele,
The alternative form of a gene.
alternative form of a gene. Very easy. Describe how artificial selection differs from natural selection.
Artificial selection takes a short time,
Yes, it does take a short time. You're correct. What else?
and, and it results in the population with a higher economic benefit.
Yes, you are correct. Okay, we will create a population with more economic benefit. Can you think of other differences?
There's less biodiversity for the artificial selection.
Yes, you are correct. There is less biodiversity. Can you talk to me about the steps?
It's the breeding of two desired organisms and utilizing their offspring.
Yes, you are correct. I just want to reach a particular point. In artificial selections, is there human interference or no human interference?
Human interference.
Yeah,
Of course, this is the most important difference. There is a human interference. Okay. In natural selection, it's exactly the opposite. It takes a number one, there is no human interference.
It takes long
It's
time.
nature here that will, yes, you are correct. It's nature here that will choose the most fit organism. It takes more time.
it
This is
results in
slower.
it results in a population that is more adapted to survival.
Yes, you are correct. More biodiverse. And here there is no human interference. Good. You answered this question very well. Mutation causes formation of new alleles, which increases genetic variation. State 2 other sources of genetic variation.
Meiosis.
Yes, you
And
are
radi-
correct. The
Random
MMRR.
fertilization.
If you notice, this question is repetitive. Random fertilization, random mating, meiosis, and mutation. Okay, mating, meiosis, mutation, random fertilization, random mating. Okay, a graph again. Scientists monitored the changes in the pH of muscles before, during, and after 2 minutes of vigorous exercise. The change in pH are caused by the production of lactic acid. Okay, very good. Again, all this is supplemental. So the pH in the muscles, obviously the pH was here, and then the pH decreased during the vigorous exercise. See, during the vigorous exercise, and then the pH went back again to normal. Okay, with time. Complete the sentences to describe and explain the results. Let's read together. The pH decreases from what to what during vigorous exercise? So I'll have to go to the graph. The pH will decrease from what to what? This is 7 and this is 7.10. And we have 10 boxes. So every box is, what's the value of every box? This is easy. 1, 2, 3, 4, 5, 7, 8, 9, 10. Now this is so easy. What's the value of every box?
0.1.
0.1.
Yes, very good, 0.1. This is exactly 7.7. So the pH will decrease from 7.07, not 7.7, until here, 7.50, 7.51, 2, 3, 4, 5, 7.55, 6.55. So they just want to know if you can read the graph. The pH will decrease from 7.07 to 6.55 during vigorous exercise. There was not enough what supplied to the muscles?
Oxygen.
Oxygen.
Very good. This is easy. The body respires anaerobically. The lactic acid produced builds up in the muscles, causing an
Oxygen.
Oxygen.
excellent oxygen debt. It takes how many minutes for the muscle pH to return to its initial level? So I have to go to the graph again. It dropped after 2 minutes and then it went back at around here. It started to become, so 30 minus 2, around 28 minutes. Or if you take it from here, 36 minus 2, that would be around 34 minutes. Or if you take it anywhere in the 30s, 34 minus 2, this will be 32 minutes. Okay, let's look at the mic scheme. This is the last question and then we will be done. Any time between 31 and 32. Okay, we are on the right track. So they want you to use the graph. The pH value increases after vigorous exercise has ended, as lactic acid is transported in the what? To the
Blood.
what?
Liver.
Blood.
Very good.
Muscle.
Transported in the blood and goes to the liver. Well done. During this time, the breathing rate and what rate remains
Heart rate.
high? Excellent.
rate.
Heart rate. Easy. Nice and easy. Okay, this doesn't finish. Yeast can respire anaerobically. Complete the balanced chemical equation for anaerobic respiration in yeast. Glucose is broken down to give me what?
2C2 H5OH.
Yes, alcohol, OH plus.
CO2.
or CO2.
Correct. Is this
2CO2.
balanced? Yes,
No.
2
2CO2.
here and 2 here. Well done. Yeast belongs to the kingdom fungus. State one cell component that is present in yeast cells but is absent in animal cells.
Vacuum.
Chitin cell wall.
Cell
Chitin cell
wall.
wall.
Cell wall. Again, this is a very easy question. In fungus, it is made up of chitin. Oh, we're finished. This is very nice. Now, this exam was easy, but you will lose a lot of questions, a lot of mistakes. You know why? Because maybe 2 questions were asking you to break down a graph and talk about investigations, and you're not good at that. Like this question out of 6 marks. I expect most of you to lose grades in this question, even though we explained it. It is clearly in your notes. But you don't know how to break down a question. I don't know why. And also this question. When you are asked to compare between two concentrations, you're always in a hurry. You always want to give a quick answer and leave. This is always going to make you lose marks. When they ask you to compare between one concentration and the other, take each one and break it down bit by bit. Write everything you know. And the same case when it comes to the graph. Look, I want to bring a whiteboard and then I'll promise I'll let you go. Because a graph will only have one of two shapes. Just give me 1 minute. Okay, now you will either have a graph that looks like this or a graph that looks like this. When you have this type of graph, which came in your mock exam, you will please divide this graph into 3 parts, part A and part B and part C. And you will discuss A, and you will discuss B, and you will discuss C. Always at B, use the term peak or maximum. This is the peak rate of reaction, for example. This is the peak temperature. This is the optimum temperature. Okay? And then you will divide it into 3 parts. So this type of graph is divided into 3 parts. This type of graph B, is divided into 2 parts. Part A, increasing part. And then part B is the constant part. Fine? And you will discuss or explain why A is increasing and why B is constant. Always when the graph is increasing at A, the limiting factor is whatever is present on the x-axis. You will have this shape or this shape. It's never any other shape. Any questions? Okay, we finished this exam. On Tuesday, I'm going to solve also another exam, 2023. And of course, don't wait for me. You have to solve on your own. And you have any questions, send me. Okay?
Okay, thank you.
Okay.
You're welcome. Bye.